So we have our basic structure, which is double-stranded DNA to be this. So this is a double-stranded DNA. As you can see, it's double-stranded and it's also in helical structure. So we have our basic structure to be this, having proteins that code for a particular inheritance or trait. So these are the proteins that code for it. And the 4 basic proteins that code for the inheritance are, I used one mnemonics for it. I used the girl ate cookies to remember these proteins. Where the D means thiamine, the girl means guanine, 8 means adenine, and C means cytosine. So this is actually a good mnemonics for this. Then, how do these proteins pair in this double-stranded DNA? So I'm going to be showing one trick for this. So the trick we have here is that the C curve proteins. So what I mean the C curve, I mean the proteins that started from C and the proteins that have curve actually pair together. So the proteins like C, which is cytosine, and guanine pair together, while T and A pair together. So we have here that if T should be here, that means adenine should be here. So they pair together. And if guanine should be here, which is G, that means cytosine is here. So this is actually the structure of the DNA. So let's move on to another structure. We're going to move into the structures to form the main structure of unit of hereditary, which is the chromosome. So now we have that, we have mentioned this as our DNA. You should take note. So this is our DNA. Now let's move to another structure. So this DNA actually, in this form, binds to a protein. This is actually the protein, the DNA. You know, this is the double-stranded DNA. Now binding to a protein, binding to this protein, and this protein is called what? Histone. And what is histone? Histone is what? A DNA binding protein, in case it comes out in exam. So histone is a DNA binding protein. So this DNA helps this double-stranded protein to bind towards it. It helps it to bind. And what is the essence of this binding? The essence of this binding is for them to be condensed, or for them to enter into the nucleus. As we know that the nucleus is very small, so it needs a little space for this our gene to be inside the nucleus, or chromosome to be inside the nucleus. So we have to make it to be condensed as possible as we can. So this DNA strand binds with these histone proteins to form what you call. So actually we have that histone plus this DNA gives us what? It gives us what you call chromatin. You guys must have been confused about chromatin, chromatid, chromosome. So actually you should listen to this, that when the DNA strand binds with the histone proteins, it gives us what? It gives us what? Chromatin. Then this chromatin we see here is the form of which the histone and the DNA bind. So this form is called the chromatin. Then let's talk about the next step before we reach the chromosome. So the next step we have is that after this binding, this actually binds to another histone protein. The same way they binds to 4 different histone proteins. So the 2 strands binds to 4 different proteins to give the octamer form, the octamer protein form. So when this binds to 4 different proteins, like 1, 2, 3, 4, at the same side, it binds to the other side, to the left hand side of the same protein. So this is actually an octamer form. It's on point 8. So the 4 proteins at one side and the other 4 proteins at the other side, binding with 2-stranded DNA structures to give all sorts octamer protein form, known as what? The nucleosome. So we can actually see that the chromatin is a condensed form of the nucleosome. So we move over to the next structure, which is the last structure, and the structure we are about to study now. So these nucleosomes, when they are in condensed form, so they condense. These nucleosomes actually condense, and they move, they condense and move and intertwine in a solenoid movement. Solenoid movement, a movement like this, like this. They intertwine together to give us what we have as our chromosome. So this is actually the structure we started from the beginning with. So they give us, they intertwine together to form this chromosome, of which chromosome has the basic unit of hereditary, which is the gene. So let's now come and study the structure of chromosome. So we have that this is our chromosome. So our chromosome, don't mind my drawing, sha. This is our chromosome. I'm going to be labeling different. These are chromosomes. Let me just label it. This is our chromosome here. And these parts of chromosome, these two parts, they are called arm of chromosome. They are called the arms of chromosome. Then we have the tip of this, of the lower arm, to be known as telomere. It's called telomere. So now we have this arm, the upper arm, to be known as what? The P arm. And we have this Q arm and we have here as Q arm. Actually here as Q arm. Remember that here is the telomere. So this tip end is called the telomere. Then what is this structure we are seeing here? This small structure at the center. We have this structure to be centromere. We have this structure to be centromere. And the centromere, what is the function of the centromere? We can say that the centromere helps in spindle. It's been involved in spindle separation when it comes to the cell division. So when the attachment of spindle is what actually happens at this centromere. So it's the division in cell division. The centromere helps in binding to the spindles for it to separate the two chromatin sisters. So and we have these two parts I'm seeing here, these two parts, we have them to be chromatid. Now see the difference now. See that the chromatin, we say that is the histone and the DNA, and the chromatid is their sister chromatids there. So these two parts, they are like, we call them sister chromatid. So these two parts are sister chromatid, D and N. There's a big difference. So chromatin and chromatid. So these two parts will be sister chromatid, and DNA plus histones to be chromatin. So this is the basic structure of the chromosome. Then I'm going to clean up this labeling. Then let's move to the types of chromosome. We have 3 basic types of chromosome, but if for additional cases you can add another one. So we have here as the centromere. I'm going to leave the centromere because it has an importance in these types we are about to discuss now. So the centromere is what actually determines a type of chromosome, because it's the point in which the centromere is determines the type of chromosome. So if the centromere should be here, if we have our centromere to be, we have our chromosome here, and we have our centromere to be here. I'll just use cent for its meaning. So if we have our centromere to be here, this is called metacentric chromosome. And why is it called metacentric? It's called metacentric because the centromere is almost to the center. I know that the centromere cannot be in the center. Let us say the centromere can slightly be in the center, because the centromere here, there's a big difference between these two arms. This is known as the short arm, and this is known as the long arm. So if the centromere is here, it actually makes this arm longer than this short arm here. So the centromere cannot be at the center, because if centromere is actually at the center, we don't have any difference in both arms, so we cannot name them short and long arms. So the centromere is slightly at the middle, or is slightly at the center. It's known as metacentric. So it's metacentric, almost at the center of the two arms. Now we have first one to be metacentric, and we label it as M. Now let's study the next type of chromosome. We have another type of chromosome when the centromere is moving towards the tip. It's moving towards the tip. So we have it as centromere here, and this is our long arm. So you can see that the centromere is almost at the tip. It's not actually almost, but slightly deviated from the center. It's slightly deviated from the center. So we have it as the sub metacentric. So it's talking about the center part. So it's called the sub metacentric. The sub metacentric is deviated from its center slightly. So this is the sub metacentric. You should know that this sub metacentric is not always included in types of chromosome, because we don't actually find the chromosome in this form most times, but we have to include it in case of any exam we are writing. Then we have the next one to be, we have written about the sub metacentric. We have the next chromosome. We have the next as the acrocentric. This is when the chromosome is almost at the peak. As you can see, this long arm is seen very well. Like we are seeing that it's very, very long compared to the short arm here. And it's because of what? The centromere is almost at the tip. And the name acrocentric here came from the Greek word, where the acro means peak. So it's actually almost at the peak of the chromosome. So we have here as the acrocentric. We denote it as A. Then the last but not the least, we have the telocentric. This one is that the centromere is already at the peak. Centromere is at the peak. Centromere is at peak. So these are different types of chromosomes we have. Like I said, the main type of chromosomes we have is MAT. So we have the types of chromosomes to be MAT, but in some cases you can include S, which is sub-metacentric. So that is all we have for types of chromosome. Then watch the next video for gene location in the chromosome and telomere explanation, like different parts I've mentioned in the chromosome, the explanation. So watch the next video for it, and don't forget to subscribe, like, and share. So also comment in the comment section if you have any information to us, or if you have any question. I will treat a video on that. If you have any question, just comment on the comment section and I'll do the video for that. Thank you and God bless you.